Well, thanks everybody for being here today. It's a real honor to be able to present some of my research, or at least portions of the key results that we've seen so far. Am I good on volume? Too loud or good? Okay. My research here does involve the use of these synthetic hexaploid wheat varieties for being able to improve drought tolerance in some of the local varieties here in Colorado, as well as throughout portions of Nebraska too. So to begin with, just an overview of what I'll be talking about today. First of all, what exactly is synthetic wheat? What does that mean to be the synthetic version of wheat? We'll talk about some of the experimental approaches and designs that we use to research this particular topic, as well as some of the results that we've seen up to this point. Some of those key results in terms of the morphology and physiology. And finally get into some of the future research that we'll look at in the upcoming steps. So let's first start out with what exactly synthetic wheat is. What does that mean? There are several different types of wheat that are grown throughout the world. The first of these, or the first major type, is durum wheat, which is a tetraploid. So that means it has two genomes, the A and the B. Bread wheat, which is common here in Colorado, and I hope you can see those colors. But bread wheat is a hexaploid, meaning that it has three genomes, the A, B, and D. So synthetic wheat is a recombination of this hexaploid variety, where we're able to take a wide range of accessions of durum wheat, where we get the A and the B genome, and cross that with many different accessions of Aegilops tauschii, which is the original progenitor of the D genome. So it's this recombination that then gives us the ability to reintroduce some of the phenotypic and genotypic variation that has been lost over the last many years, I guess, of wheat breeding that gives us the type of wheat, bread wheat, that we see today. And you've seen this picture before from Pat's earlier talk about the comparison between local bread wheat as well as that original progenitor, and what we're kind of having to deal with in terms of getting the good traits out of this Aegilops tauschii. So we'll move on into the experimental design and what some of the things we did to research the feasibility of synthetic wheat. So we started out with 400 different lines of synthetic varieties that were produced by Simmet. From within those 400 lines, we were able to narrow this down to 6 different synthetics that we chose just basically from agronomic aspects, or the visual traits that we thought would be good for use here in Colorado for improving drought tolerance. And we ended up with these 6 different lines here, just arbitrarily numbered. Each of those were then crossed and back-crossed to the 2 local cultivars, Hatcher and Goodstreak, which are locally adapted to Colorado and Nebraska. So these 12 different populations that were created from this were then tested in the field, 8 different environments for yield and drought tolerance through Colorado and Nebraska. And from this, we actually saw some slight improvement. Just a slight improvement in yield for those back-cross derived families, a few of them, in terms of their relation in yield to some of those recurrent parents. So there was some, I guess, some cautious optimistic about what we've seen. So next we moved on into trying to identify what particular traits contributed to this improvement in yield, or that very slight, mind you, the improvement in yield. So we developed this root column study where we analyzed the 6 synthetics that we chose, as well as 4 local cultivars, Hatcher and Goodstreak, the recurrent parent in the back cross lines, as well as Ripper and Bird, which are 2 of the most highly productive varieties that are found here in Colorado. So we had a split plot design with 6 different reps, 2 treatments. So we had a well-watered treatment, which was just saturated on a daily basis, and our drought treatment just consisted of removal of irrigation after 10 days post-emergence. that wasn't necessarily an abrupt stop of water, because it actually spanned a 3-week period of that dry down, because we were able to use these 1-meter long tubes. So we had the soil profile primed with water, and the roots were actually able to go down and search out the moisture through those tubes. And another thing that I should mention about this too is the soil media that we used, or the type of growing material, was a fritted clay. So it's a clay-based material that had a lot of the water holding capacity of a pretty decent soil type, but it was the consistency of sand. So it was really easy to, while we were measuring these roots at harvest, it was easy to wash these roots free from the soil. And from that, we eliminated a lot of the confounding factors that would go along with a typical root study. So the measurements that we took we harvested just prior to the shift to the reproductive stage of the plant growth. So we measured this during the vegetative growth. We measured a wide variety of different phenotypic and physiological and morphological measurements, some of which you see here. But our primary goal or interest was this root morphology. That's really what we were after because of its contribution to drought tolerance. And to do that, we separated the roots in these tubes into thirds, the top, middle, and bottom third of those tubes, and analyzed the roots within each section with this software program called WinRhizo. And I'll talk about that briefly, but this is just a software program that allows us to take a digital image of our root system after it's washed free from the soil. And from that, we can get many different traits, primarily the diameter of the roots, the root length within certain diameters, surface area, different measurements such as that, which otherwise would be almost impossible to measure by hand. Okay, so let's move on into the results that we actually saw, some of the key results that we found in this initial research. So we started out with wanting to be able to classify our varieties in terms of whether they could be actually considered drought tolerant or not. So we used the relative water content of the plants, or rather the percent reduction in relative water content that was found under that dry condition or the drought stress condition to classify whether these particular lines were tolerant. So we identified 3 varieties that showed the largest percent reduction in relative water content. We termed them our sensitive group. And of course, the 3 varieties that were the lowest in loss of relative water content, those were our tolerant group. So being able to have this grouping system like this then allowed us to look at all of our other traits we measured and compare them to these two traits to see if there's any significant difference between them, and to give us some indication of whether or not all of the other traits may contribute to the separation in these groups. So we'll refer to this grouping a little bit throughout the rest of the slides. So looking at one of the key traits that we wanted to test in this research, the production of root biomass. And from this graph, you can see that we again break it down into our groups, sensitive versus tolerant varieties. And we have root biomass on our y-axis, and down here at the bottom is our root depth. We'll get to that in just a second. But first of all, the root biomass that was produced at the top, middle, and bottom thirds of those tubes, broken down into the different groups, those different sensitive versus tolerant varieties, and you see a significant difference for both treatments within the middle and the bottom root biomass section of those tubes. So that really indicates that there is a good contribution of rooting capacity to being able to separate these 2 groups, the sensitive versus tolerant varieties. And of course, with root depth that has been talked a lot about in literature and other things too, we again see that separation under both treatments to indicate the capacity of these tolerant varieties to produce more vigorous roots even under those stress conditions. So the next thing we wanted to do was try to identify where this change in biomass was coming from. So we analyzed the difference in root growth in terms of root length. And I'll walk you through this a little bit. This tries to illustrate the difference, or the data that we collected from that WinRhizo software program. Okay, so from within this graph, we see our top, middle, and bottom third of those tubes that are separated, and the roots within them, of course, relate to those sections. The root size in the diagram relates to the actual diagram class that was measured by WinRhizo, and those classes relate to the size of the roots. So as you move down through our size classes, hopefully you can see those roots at the bottom too, but the size of the roots in the image relates to those classes. The length of the root is indicating the difference in growth for that particular diameter class, as it differed between the sensitive versus tolerant group. So for example, this root, that largest diameter root at the top, produced 50 millimeters more roots in terms of length than under the sensitive treatment, or the sensitive varieties, as compared to the tolerant varieties. Hopefully everybody was able to catch that. So when we're looking at the difference between the irrigated and the stress treatment, we began to see a little bit of a pattern emerging in the irrigated treatment where the sensitive varieties were producing larger roots. We see that from that top, the largest diameter class, the 1 millimeter and above, showing a larger production of roots under sensitive, for those sensitive varieties. What we're interested though is how those roots respond under stress treatments. So under tolerant, the tolerant varieties under the stress treatment, we begin to see a pattern of producing the smaller roots. I hope you guys can see that, but there's a large difference in production of root length for the tolerant varieties in these very small diameter root classes, which indicates the importance of being able to produce a smaller root that can cover more areas. So it's kind of like what weighs more, a ton of bricks or a ton of feathers, and it relates to this. Being able to produce biomass that can grow out and expand to a larger soil profile, can capture more water and maintain those levels of water status in the crop. All right, so let's move on into some of the above ground traits that we measured. First of all, we had the stomatal traits. We measured stomatal conductance through the use of a leaf perometer. And we also took measurements on stomatal density as well as aperture. The density, some of the pictures that you see here, didn't really give us a significant difference between the two groups. It did on a genotypic basis though, so I still wanted to show you what that looked like and keep that, give you an image in your mind. For the stomatal conductance, however, we did see a significant difference for the tolerant varieties showing a higher conductance level than the sensitive varieties under that drought stress. So that's exactly what we want to see, right? We want to be able to see those varieties that can maintain a higher level of water status, still be able to maintain conductance as well, rather than showing the mechanism of essentially dormancy, or being able to maintain a higher level of water status through the plant just shutting down. So this conductance level is a good indication that that's what we want to see. For the stomatal aperture trait, we saw something that we didn't really expect, in that tolerant varieties showed larger apertures under both conditions. And we don't expect to see that because it's been reported that a smaller aperture size may contribute to improvements in drought. So a smaller aperture means smaller guard cells. And a smaller guard cell has been indicated as having a faster response time. So from that, a smaller guard cell should be able to minimize unnecessary water loss through the stomates. It may be that the reduction in leaf growth under drought conditions, where these sensitive cultivars didn't have the water status to continue cell growth, that those would show a smaller aperture size. But because we see that in both wet conditions, that tolerant varieties are larger in both cases. There's definitely something genetic behind that is indicating that somehow the aperture does relate to an improvement in drought. We'll talk about the measurement of osmotic adjustment next. We have the relative water content on our y-axis and osmotic potential on the x-axis. And I want to point out too that the symbols that are associated to these different varieties, for example, the orange triangle relates to synthetic variety number 194, and it's those symbols that indicate the well-watered treatment. So the dots on the other side of the line that's our drought-stressed side of this line. And from what we see here, these 4 different varieties are actually related to are indicating the 4 varieties that were our highest producing varieties. Those aren't necessarily our tolerant group. Those are the highest level of production that we saw in yield from our field studies in previous years. So when I bring in the next 4 varieties, we see a pretty good significant reduction in relative water content, as well as a slight decrease or an increase in those negative values of osmotic potential. So the lower the osmotic potential, the more capacity of that cell to intake water, right? So there may be several different things that can explain or at least give you an idea of what's going on here, why we see a reduction in relative water content and a decrease in osmotic potential. One of these would be that the reduction in relative water content or the loss of water could just contribute to a passive increase in osmotic potential. Another option is that the reduction or slight reduction in osmotic potential down here to these more negative numbers actually contributes to some cell rupture during the relative water content sampling process. So that greatly reduces our amounts of relative water content. But looking into this a little deeper, we may be able to identify a second mechanism or an additional mechanism that can contribute to this reduction in relative water content. So these numerical values are actually the rank for the production of root biomass in our bottom section of those tubes. So rank number 1 produced the most biomass at depth throughout these different, throughout the different tubes. And the next group that showed our low values of relative water content were the smallest producers of root biomass, again indicating the importance of those roots to be able to go out and capture the root, the water wherever it is available. So moving on, what we'll do next with this research in some of the future experiments, we want to be able to validate some of the things that we've seen. It was in terms of, do the synthetic varieties show similar results in an actual field setting? We want to be able to also test those backcross derived families in terms of the same thing. What types of traits are they contributing to? We will be able to select certain backcross families and genotype those to be able to identify what synthetic regions may be maintained to be able to contribute to those yield improvements that we saw. So just in conclusion, the synthetic varieties that we did test, certain lines did improve overall yield to a slight degree, And there is a potential for improved productivity under drought as well. We pointed out that the capacity of the roots is one of the key functions to be able to capture soil moisture, which is, I think, fairly well known in the industry. But what we want to do is to be able to identify if certain molecular research needs to be done for identifying regions within that synthetic cultivar for this improvement that we saw. So we'll be able to utilize that to identify these molecular regions for efficient integration into future cultivar research. So with that, thank you very much. I'd like to thank a few people too that helped me through all of this research, mainly my committee members, Pat Byrne, Scott Haley, Bill Bowerly, and John McKay, as well as Steve Benzinger for his donation of some seed and efforts, as well as Scott Reed and Judy Harrington, who were invaluable in this research, as well as, last but not least, my fellow graduate students and Byrne lab workers who put in many hours sampling physiological measurements. So with that, thank you very much. I think we probably have a little bit of time for some questions.